Welcome everyone to our first lecture on Chapter 1, An Introduction to Biology. Before we get started, because this is our first lecture, I want to remind you to have a separate sheet of paper handy or a Word document open on your computer so that you can answer the checkpoint questions as they come up throughout the lecture. And then later when you finish the lecture, the answers that you come up with to those questions will be submitted on Blackboard. So, the goal of this lecture is to dip our feet into the subject matter of our class by introducing you to biology as a science. You probably already know that biology is the science that studies living organisms. And that might seem like a pretty simple explanation, but we need to dig into it more. And so in this lecture, we are going to take an approach to understanding biology by exploring two questions, which are, what is science? And what does it mean to be alive? And we're actually going to start with the second question first. What does it mean to be alive? What makes a living thing? What are the characteristics of the things that fall under this subject of biology? And this might sound like an easy enough question to answer, but if you take a look at the pictures that I have just put up on the slide, everything pictured here is alive. From plants, to animals, to humans, to tapeworms, to the mold that grows on a loaf of bread, to microscopic organisms like bacteria. Despite how different these things all are from each other, everything that you see here is alive. But since they're all so different, what is it that unites them? What is it they share in common? And what is it that distinguishes them from non-living things? So you might be able to think of several things they have in common off the top of your head, but what biologists recognize as the defining qualities of living organisms include 7 different properties. And these properties shared by living organisms include order, sensitivity or response to stimuli, reproduction, adaptation through evolution, growth and development, regulation/homeostasis, and energy processing. For some of these, you can probably gather what they mean just from looking at the word. For others, maybe not. So let's briefly take a look at what each one means, starting at the top of the list with order. So one characteristic of living things is that they are highly ordered, or in other words, organized in their composition. They're made of certain types of molecules, and those molecules are organized into very tiny structures called organelles, which perform certain functions inside of cells. So all living things are made of cells. And in some organisms, the level of organization stops there because some organisms are made of just a single cell. Bacteria are one example of a living thing made of just one cell. However, in larger multicellular organisms, cells are organized into tissues. So for example, human skin or muscle are both tissues, but tissues are also found in plants. For example, plants have dermal tissue, which is like their skin, which covers the plant and protects it. In multicellular organisms, tissues are then organized into organs and organ systems like our heart, lungs, nervous system, and then those organs are found nested within the organisms. And a large part of what we're going to be exploring in the first few weeks of our class are these first few levels of organization in living things, the molecules that they're made of, their organelles, and cells. Moving on, the second characteristic that living organisms all share is sensitivity or response to stimuli. So living things are sensitive to signals that they detect in their environment, and they respond to those signals. This is true for everything from the largest land animals to the tiniest microscopic organisms. So when your cat bounces off the walls after being startled, that is a response to a stimulus. When a sunflower tracks the path of the sun across the sky throughout the day, like what you see in this time lapse, that is a response to a stimulus. Even little bacteria, which we can only see under a microscope, can often move around and swim toward a particle of food, like what you see happening in this video here. So very different organisms, but they're all responding to signals in their environment. The third characteristic is reproduction. All living things can produce more of their own kind. And again, this includes animals, this includes plants, and this includes microscopic organisms. In the middle, there is a time lapse of bacteria multiplying under the microscope. All living things make more of themselves. The next characteristic is adaptation through evolution. Now, your textbook actually counts these as two separate characteristics. It counts one as adaptation, the other as evolution. But in reality, they are more like one characteristic, and that's how we're going to choose to treat them. Because the meaning of adaptation here is not the colloquial use of the word adaptation. When we use the word adaptation in everyday language, we usually mean that an individual is making a change to overcome a situation. So for example, if I find that my exercise routine isn't producing the results that I want, then I might adapt and try a new approach. And that is one definition of adaptation, but that's not the biology definition of adaptation. The biology definition of adaptation is where over the course of many generations, groups of organisms develop features that are encoded in their genes that enhance their survival in a particular environment. For example, an Arctic fox's white coat is an adaptation that provides it with camouflage in its environment, which is something that provides it with a survival advantage. Saguaro cacti have special mechanisms for conserving water in the dry desert climate that is their natural habitat. And both of these adaptations, along with the countless others that you see in the biological world, have evolved over many, many generations to suit the survival needs of that organism. But those survival needs are not always a good thing for humans. For example, a major area where evolution is impacting human medicine nowadays is the evolution of adaptations in bacteria that make them resistant to antibiotics. So antibiotics are the medications that are used to treat bacterial infections. And over the decades that the medical field has been using antibiotics, the proportion of bacteria that are resistant, non-responsive to common antibiotics has increased significantly. Because for bacteria, being resistant to antibiotics is a survival adaptation for them. Although for us as humans, their survival would be a bad thing in this context. And this is something we'll discuss more later, but hopefully you can see from this brief discussion how all manner of organisms evolve, and that leads to adaptations. The next property is growth and development. This one's pretty straightforward. All living things grow, mature, and develop traits encoded in genes that were passed down to them from their parent organisms. Next is regulation and homeostasis. So all living things will coordinate and regulate their functions. And part of the goal of this regulation is to maintain something called homeostasis, which is the scientific term for stable internal conditions. So as living things, we have to maintain internal conditions that are suitable for sustaining our livelihood. And this can include body temperature, it can include pH, water content, concentration of important materials in the body. For instance, one way that humans maintain the temperature as part of their homeostasis, which I'm sure we've all experienced, is sweating. Sweat is a response to elevated body temperatures, and it's meant to keep the body cool. Another less obvious but still very important way that our body regulates itself is by maintaining normal levels of blood sugar. The body uses a pretty complex system of hormones and signals to ensure that blood glucose levels don't get too high or too low, and that they stay right in a safe and healthy range in order to be able to feed your cells the energy that they need. We can also even see regulatory processes in microscopic organisms, like this paramecium that you see on the right-hand side of the slide. Paramecia are microscopic single-celled organisms that live in ponds, streams. You can find them in most watery habitats. And they have a special structure inside of them called a contractile vacuole. You can see it in action here, which will expel excess water that the paramecium takes on and helps it maintain homeostasis by keeping its water content in balance. So you can see that contractile vacuole expanding and then contracting down as the water gets pushed out of the cell. So every living thing has to regulate itself and maintain homeostasis, whether it's a large organism or a small one. Our seventh And final characteristic of living organisms is energy processing. So all organisms need a source of energy to go about their normal business, and this varies from species to species. We, of course, consume food as our source of energy, but plants obtain their energy from sunlight. Microscopic organisms use a wide range of different processes. Some use the energy of sunlight, others eat, very much like we do, but they, of course, aren't eating hamburgers. They instead eat little food particles that are even smaller than they are. In this video, we can see a cell chasing down and engulfing a particle that it wants to break down. And several weeks from now, we will be looking at a couple of the main metabolic processes that organisms use to obtain and process their energy sources. So this brings us to our first checkpoint question. So remember, this is one of the questions that you will be turning in on Blackboard, so be prepared to pause the lecture, come up with your answer, and then write it down or type it up somewhere before proceeding. So during the Voyager missions to Mars, which took place in the 1970s, an experiment was conducted on samples of Martian soil that showed some indication of energy processing happening within the soil. Does this mean the Voyager missions proved that there are microscopic organisms living in the soil on Mars? Why or why not? So with that, we have come to the end of our discussion of the question of what does it mean to be alive? And now we're going to transition to the other question that we are exploring in this lecture, which is what is science? And just like with the question of what it means to be alive, there's more to this question than meets the eye. And over the years, I personally have read a lot of different definitions of science from many different sources. And no two sources are exactly the same, but at the core of those definitions are a few features. And those features are the use of evidence and the use of reasoning. So we will define science for our purposes as a very specific way of learning or knowing about the world by using evidence and reasoning. Now, what do we mean by evidence and reasoning? Evidence is real-world information, facts, or data, observable information that you can collect about the natural world. Reasoning is the thought process of coming to logical conclusions. And while this might sound kind of formal in nature, one of the things that I really want to impress upon you guys is that we all do a less rigorous version of science every single day, simply as we move through the world making decisions and trying to solve problems. So for example, let's say that you are faced with a situation where you are feeling jittery in the evening and you can't fall asleep. Why might this be the case? Well, let's say you drank coffee at 4 p.m. that day. The fact that you drank coffee is your evidence, and your reasoning is then coming to the conclusion that drinking coffee in the later afternoon makes you feel jittery and is a bad idea if you want to easily fall asleep. What you just did there is science light, quote unquote. You used evidence and reasoning to solve a problem and answer a question. And science is just like that, only it has more rules for how it operates. So in science, evidence has to be gathered a certain way to ensure that your conclusions are reliable. So let's take a look at how a scientist might approach this very same problem. So a scientist might recruit 500 participants for a study. Half could be asked to drink 8 ounces of coffee at 4 p.m. every day for 1 week. The other half could be asked to not drink coffee at 4 p.m. And then the participants could be asked to complete a survey every night at 9 p.m., rating their jitteriness on a scale of 1 to 10. And then after a week of this survey, you take the scores from the coffee drinking group and compare them to the survey scores of the non-coffee drinking group. And then let's say the average score in the coffee group is 3 points higher than the group that didn't drink coffee. Now, based on this evidence, we can reason that drinking 8 ounces of coffee at 4 p.m. causes people to become more jittery at 9 p.m. So as we can see, the scientist's approach to answering this question is more rigorous and it's more regimented. It includes observations of many people who are all doing the same thing at the same time, and there's this standardized way of collecting the results through a survey that allows you to quantify it. And we'll talk in more detail about why it is important that scientific studies have this sort of regimented structure with these features. But before we get to that, I have another checkpoint for you where I would like you to reflect on a specific instance where you have used evidence and reasoning in your everyday life this past week. It can be something completely mundane, like noticing that you are jittery after drinking coffee, or it could relate to solving a big problem in your life. So please tell me what you noticed, which represents your evidence, and what you decided and why, which represents your reasoning to come to a conclusion. So before we continue, it's also helpful to look at the definition of science not only from the perspective of how does it operate, but also what kinds of things are not part of the operations of science. What is excluded from science? One of these things is opinions or feelings, meaning judgments or viewpoints that are not necessarily based in evidence. For example, if your favorite color is green, there's not really any evidence to support that. It's just how you feel. If pizza is your favorite food, again, that's just a personal opinion. So science doesn't do that. Another thing that science doesn't do is supernatural or paranormal matters, which are by definition matters related to forces that are separate from the observable natural world. And given that science, again by definition, looks at observable, verifiable facts and evidence, and supernatural things are by definition separate from the natural observable world, science does not study those things. So things like ghosts, fairies, mythical creatures, science looks at phenomena that are observable, detectable, things where there's verifiable evidence. And despite what you may have seen on TV shows like Ghost Hunters, these supernatural things do not fall into that category. And I also want to use this lecture as an opportunity to talk about the importance of science literacy. Why is it important, even for people who are not scientists, to know how science operates? Even if you have no interest in going into a scientific field, understanding how science works, how studies are conducted, how they produce results, all of this is really important because science plays a major role in your daily life. So much of what we interact with on a daily basis is the product of scientific studies. Everything from the food that we eat, the medications that we take, and other ways that we care for our health, the air that we breathe, the ecosystems that provide us with energy and resources, the technologies that we use. These are all ways that science is with you constantly as you go about your life, even if you don't realize it. So every time you eat a vegetable, you are eating decades of research. Every time you take a medication, even an over-the-counter one, you are taking millions of dollars of testing and clinical trials that has ensured that that medication is safe and effective. So science enables us to live our lives the way that we do today, even if we take that fact for granted most of the time. But another reason why it is important to understand how science works is so that you can be informed and empowered to know when the truth is being communicated to you or not. Because the unfortunate reality is that the news does not always get it right. When scientists make a discovery and the media communicates that discovery, sometimes, not always, but sometimes, it is totally lost in translation. And sometimes this is because journalists don't really understand the research. And sometimes, unfortunately, it's because the research doesn't make for a very exciting headline. And so to grab more eyes, they stretch the truth. But whatever the case, it's important that you have the tools to understand when the truth is being communicated correctly or is being misrepresented. And I'll give you one example. So in the study that we see, this screen clipping on the left here, which is entitled Experience with Anonymous Interactions Reduces Intuitive Cooperation, scientists, specifically behavioral psychologists, set up a laboratory experiment where participants were asked to play a video game with an anonymous stranger who was in a separate private room, so a person that they did not know and could not see. And they found that over time, the participants would become less cooperative with each other in the video game, given that they believed that they would never have to interact with that person again. Now, when the press release on this study was put out by the media, here is the headline. Is big city living eroding our nice instinct? Okay, whoa, that's kind of different. So this headline and even the contents of the article itself might lead you to believe that the researchers looked at how nice people are who live in cities versus how nice people are who live in rural areas, and that people in cities are not as nice. However, that's not at all what the researchers did. And in fact, their findings had nothing to do with city life. The journalists are just making these extrapolations to spice things up. And you would never know that unless you clicked on the link to the original article, read the summary, and realized that all of this stuff had been lost in translation. So it's important to have science literacy so that you can understand what's real and what isn't when science info is being communicated to you through a middleman like the media, which is almost always how it's communicated. And lastly, it's important to understand how science works, not just because the communication of science can be flawed, but because the science itself can be flawed. Science does not exist in a vacuum. It is done by people, and people can be fallible. And to be clear, as I've said, science has produced amazing improvements in our lives, and most people do believe that science has done more good than harm in the world. It's doubled human life expectancy around the world. It's created technologies that allow us to live in comfort. It's allowed us to learn about ourselves and the natural world around us. But science has also made mistakes. Science has been, in the past and now, unethical, racist, sexist, and just plain incorrect on many occasions. And it's important that we recognize these realities and not shy away from them. So, for example, our model of the solar system used to be geocentric, meaning it used to be thought that the Earth was at the center of our solar system. And now we know that was wrong. The Earth is not at the center of the solar system. The sun is. And over time, that mistake was corrected with scientific progress. But some mistakes cannot be corrected, especially those of an ethical nature. For example, many of the important life-saving procedures in obstetrics and gynecology that are still used today were developed through experimentation on Black women who were enslaved in America. And over time, ethical conduct has gotten better in the sciences, and today researchers are beholden to maintain certain ethical standards. But we can't ignore the fact that bad science has been done. Even in modern times, there are cases of bad science. So in 2021, for example, the FDA chose to approve a new medication to treat Alzheimer's, even after 0 out of the 11 doctors and researchers on the advisory committee voted for its approval. Not a single one. And the reason why none of them voted for it was because there simply wasn't enough evidence that the medication has any effect at all on improving the symptoms of Alzheimer's disease. But despite this, the FDA steamrolled their opinion and approved the med. And as a result of the FDA's decision, several members of the committee ended up resigning. So the point is that even today, there's cases of science being done badly. And the bottom line is that it's important that you, as an individual, have the knowledge and tools to be able to evaluate what counts as good science versus bad science so that you can make the best decisions possible about how to live your life. And to be clear, my message to you is not that science as a whole is bad, because on the whole, science does make our lives better. But we can still believe in the power of science while also making sure to be diligent about understanding its points of failure. And one of the goals of this class is to help you build those critical thinking skills. Now, everything that I've just said may give you the impression that there is good science and then there's bad science. But really, the situation is more nuanced than that. The quality of most scientific research lies on a spectrum, rather than being black and white, good or bad. So one feature that improves the quality of a study is having a large sample size. Studies with large sample sizes are better than small sample sizes. Sample size is the term for the number of participants included in a study, or number of observations that are made. A larger sample size gets you a better, more reliable result. For example, if we were to ask the question of what is the average height of a human adult, which of these would give you a better answer? Measuring the height of every student in our class, or measuring the height of every student at Central Arizona College? Most of us can automatically recognize that measuring every student at CAC is better, and that's the option with the larger sample size. Because what if our class just happened to be filled with a lot of short people or a lot of really tall people? The smaller the sample size, the greater chance you have of outliers skewing the data. So larger sample sizes are better. They get you closer to the truth of the answer. Another feature of good studies is having a well-balanced sample, not just a large one, but one that has good representation of the whole breadth of what you want to study. For example, in the previous example, asking the question of what is the average height of an adult human, if we decided to only include males in our sample and exclude females, you would get a very different answer because you don't have a well-balanced sample. So in human studies, the best studies have a diverse range of participants. Historically, this has unfortunately not been the case in a lot of medical research. And for a long time, clinical trials were only open to men and women were excluded, which of course leads to problems because the male and female anatomy and physiology differ in some significant ways. And failing to consider how the female body responds to medicines and treatments can lead to big gaps in knowledge. And this issue has improved over time, not only with the inclusion of different genders, but also inclusion of people with different racial and ethnic backgrounds. So for example, if you look at the data for the clinical trials of the Pfizer COVID-19 vaccine, the distribution of participants coming from different races and ethnicities matched the distribution of the whole US population fairly well. So in the middle column of this table, you can see the percentage of each group in the US population based on census data. And then in the right-handmost column, you can see the percentage of each group that was included in the Pfizer vaccine trial, which by the way included over 44,000 participants. So it was a very large sample size. For example, 5.9% of the US population is Asian, and 4.4% of the clinical trial participants were Asian. So not perfect, but a decent match. And so in this particular example, this clinical trial was considered to be a success in terms of having a well-balanced sample. Another area where there is a spectrum of quality is on the type of study. So in general, experimental studies are better than observational studies because experimental studies give you more conclusive results. And this point requires a little bit of elaboration. So let's take a look at the definitions of these two types of studies side by side. In an experimental study, the researcher is the orchestrator who sets things up. The researcher intervenes to set up certain conditions and determine their effects. On the other hand, in an observational study, the researcher observes a phenomenon but does not intervene with what they are observing. They collect data, but they don't interfere to set up any conditions deliberately. And before we go any further, now that we know the difference between an experimental and observational study, I would like you to apply that knowledge in this checkpoint by sorting this list of hypothetical studies into one of the two categories. Is each one observational, or is it experimental? So please read each example and then create a list of the observational studies and then a second list of the experimental studies. So in other words, if you think A, B, and C are observational, then you'll list A, B, and C as the observational ones, and then the other ones would be experimental. And I will give you a hint. There are 3 studies of each type, so 3 in each category. Okay, so now we can hopefully tell the difference between experimental and observational studies. But why is it that experimental studies tend to be better than observational studies? Well, a good experimental study can establish something called causation. That is, when you can conclude that one thing causes another. An observational study cannot establish causation. It can only show something called correlation, which is when 2 things tend to happen together. And this distinction is nuanced, but it's important. Correlation and causation are not the same thing. Observational studies can show you that 2 things tend to occur together. However, this does not always mean that one thing causes the other. For example, this graph shows the number of people who drowned by falling into a pool over a 10-year period between 1999 and 2009. So the number of people who drowned by falling into a pool is shown in the red line. The graph also shows the number of films that Nicolas Cage starred in over that same period of time. And that's shown in the black line. And if you look at the red line and the black line, you can see that these 2 things correlate with each other decently well. They track each other. When Nicolas Cage's movies go up, so do pool drownings. And when his movies go down, so do the drownings. But obviously, people drowning does not cause Nicolas Cage to star in movies or vice versa. The bottom line is that when all you're doing is making observations and collecting data without setting up controlled experimental conditions, then you can see that 2 things track with each other, but you can't really say that one thing causes the other thing. And that was a very silly example, but let's take a look at a real example. So there was a study, an observational study, that found that babies who were breastfed grow up to have a slightly higher IQ on average than babies who are fed formula. And at first glance, the result might seem to suggest that being breastfed causes you to have a higher IQ. But we cannot conclude this. Why not? Well, mothers who breastfeed tend to be richer and have a higher level of education than mothers who do not. These may be the reason for their children having a higher IQ rather than the breastfeeding. So it may not be the case that breastfeeding makes your child more intelligent, but rather having money, education, and resources allows women to free up the time needed to breastfeed. And because of these same resources, these children might also live in more well-funded school districts that foster their education, leading to a higher IQ. So the 2 things may occur together, but that doesn't mean that one caused the other. Now, if you wanted to set up an experiment on this situation and establish causation to truly come to a conclusion, what you would have to do is recruit a large sample of women to participate in the study. And your sample would need to be balanced and include women from all ends of the income spectrum and all ends of the education spectrum, and then divide them into 2 groups and say to them, you ladies in group 1 are going to breastfeed your children, and you ladies in group 2 are going to give your babies formula. And then compare the IQ of the children when they get older. But as you can imagine, this would be very unethical because how people choose to take care of their babies is a very personal choice. And people simply cannot be forced to give formula or to breastfeed. So in situations like these, this is why we have observational studies, because it would not be ethical to perform an experiment, even though it might lead to a stronger conclusion. So experimental studies lead to stronger conclusions than observational studies, but they aren't always possible. Moving on to another feature of good scientific studies, experiments that include something called a control are better than ones that do not include a control. A control is a test that is expected to give a certain known result, but is included in a study to serve as a comparison for the results of the experiment, or to show that the experimental setup is working properly. When we're looking at human studies, the control group is often referred to as a term that you might be more familiar with, the placebo group. So when a study is experimental, it's important that a control is incorporated into that experiment. Let's see what that looks like in a few different situations. So let's say in a clinical trial of a new medication, half of the participants are given the medication, while the other half are given a pill that contains no active ingredient called a placebo, and the results of the two groups are compared. And this is important because there is a psychological component to medicine where if people merely think that they are taking something that will help them, then their symptoms can improve. So it's important to distinguish the psychological component from the true medical component by including a control group. Now, what if the placebo group shows results that are just as good as the medication treatment group? So let's say both groups experience a 20% improvement in their symptoms. That would mean that the medication isn't actually doing anything. It's just something called the placebo effect, which means that the improvement is simply a consequence of people thinking that they are getting a medication that's making them better. Here is another example. So let's say a new variety of wheat is tested for drought tolerance by being grown under low water conditions. And then in an adjacent plot, the same variety of wheat is grown under normal water conditions. The normal water plot of wheat is your control. So why would the researchers do this? Well, what if both plots of wheat die? This then tells you that the problem was not the water level, but something else. Maybe both plots of wheat were infected with a blight. Maybe there was heavy metal contamination in the soil. Without the control group grown under the normal water conditions, you might assume that it was the low water that was killing the wheat in the first plot. And therefore, you would come to an incorrect conclusion that the wheat is not drought tolerant when in reality, something else must be going on. And then I'll give you one more example. Say 3 samples from a crime scene are collected in tubes of saline, in other words, salt water, and then analyzed to determine whether there is any DNA present. And then an empty tube of saline is also analyzed, and that empty tube represents your control. Now, what if the empty tube comes back positive for DNA? Well, that would mean that there was cross-contamination. If DNA somehow got into the tube of pure saline, maybe it got into the other tubes as well from some other source. And then maybe that means it wasn't truly present at the crime scene. So this tells you that something has gone wrong in your collection process before running the test. And that's the value of having the control in this situation. The last item on our list of features of good quality science is that studies where the researcher has no conflict of interest are better than studies where the researcher does have a conflict of interest. A conflict of interest is defined as a situation where the researcher stands to benefit from achieving a certain result in their research. For example, let's say that a researcher is testing a new treatment for a hair loss, and that researcher owns stock in the company that manufactures the treatment. The researcher has a conflict of interest because they have an external incentive to come up with a good result because it would benefit them financially for the company that they own stock in to do well. So we've learned a lot now about the qualities that make research good. And in this checkpoint, I would like you to put what you have learned into action by making your own assessment about which of these scenarios shows the markings of good quality research. So I'd like you to make a list of these items, A through E, starting with the one that represents the strongest, highest quality evidence, and then make your way down to the weakest, lowest quality evidence. And as you're reading through them, think about the features of good quality research that we just covered and use those to assess how these stack up against each other. Now, without fully giving away the answer to that checkpoint, I do want to discuss with you the comparison between two of these hypothetical studies, the one shown in letter B and D. So B describes an experimental study of 3,000 diabetes patients where half are randomly assigned to take insulin and the other half are randomly assigned to take Ozempic. And at the end, they find no difference between the health of the two groups. D describes an observational study of 3,000 diabetes patients, so a large sample size that's the same as in letter B, and finds that people who take Ozempic have better outcomes than those who take insulin at the end of the study. So the conclusions of these are saying different things. B is saying there's no difference. D is saying Ozempic is better. So which one are we supposed to believe? Well, B is better than D. B is more reliable. And that's because it's an experimental study, while D is an observational study. Remember, observational studies show correlation, but not causation. There are reasons other than their choice of medication why people who take Ozempic might be doing better than people who take insulin. So for example, Ozempic is much more expensive than insulin. And in general, people who have a higher income tend to be in better health than people who have a lower income because again, like with the breastfeeding study, they have more resources. So it may be the case that people who take Ozempic also happen to have more money to buy healthy food, invest in exercise equipment, whatever it is, compared to the people who take insulin, rather than Ozempic being the true cause of the better health outcomes. So the better health outcomes in the Ozempic group might be related to their income, not to the effect of the Ozempic. So again, like we mentioned before, experiments give you a higher level of control to eliminate some of those sources of uncertainty and truly allow you to say that this thing causes this other thing. So experiments allow you to control what are known as variables, which are defined as anything that can change in a study. But in an experiment, there are two types of variables that we do want to change. One of these is called the independent variable, and that is something that the experimenter alters by design. Then there is the dependent variable, which is the outcome of the experiment that is measured. And they're named this way because the dependent variable depends upon the independent variable. So let's look at this in the context of an example. Let's say a scientist wanted to determine the ideal amount of fertilizer to maximize crop yields on a commercial farm. The independent variable would be the amount of fertilizer used. They would set up different plots and they would give different amounts of fertilizer to each plot to find the ideal amount. Then the dependent variable would be the yield of the crop from each plot, which is the outcome that's being measured. So importantly, the yield depends upon the type of fertilizer. Dependent depends upon independent. When you're trying to identify the independent and dependent variable in a scenario, you want to ask yourself, what causes what here? Does the crop yield cause the amount of fertilizer to change? No, the amount of fertilizer causes the crop yield to change. And that's how you can pick out that dependent is the yield, independent is the fertilizer. So those are the variables that should change in an experiment, the ones that should vary. Ideally, everything else would be kept constant by the experimenter to the greatest extent possible. Why are the other variables kept constant? Well, let's revisit this example of trying to determine the ideal amount of fertilizer to maximize crop yields. If we truly want to isolate the effects of the fertilizer, we don't want any other variables to enter the picture and complicate the conclusions. So in an experiment like this, you would want to keep all of the other factors constant across the different test plots, including the amount of sunlight that they are getting, the amount of water that they're getting, the quality of the soil found beneath them, which and how many herbicides and pesticides are used, and everything else about the treatment of the plots. If these things are not kept constant, so say, for example, one plot gets more hours of shade than another, then you can't truly say that differences in the crop yields are caused by differences in the fertilizer, because you have other variables that also might be influencing the outcome. So that's why it's important to keep every variable other than the independent and dependent constant to the greatest degree possible. So here we arrive at another checkpoint, or rather series of checkpoints, where I'm going to have you apply these principles to a new experimental scenario. So an experimental study is conducted to determine whether eating yogurt can help improve the symptoms of irritable bowel syndrome. One group of participants is asked to eat 2 servings of yogurt per day, another group is asked to eat 1 serving of yogurt per day, and a third group is asked to refrain from eating any yogurt. All groups complete daily surveys reporting the severity of their IBS, irritable bowel syndrome symptoms. So in this experiment, I'd like you to tell me, what is the independent variable, what is the dependent variable, and what is the control group? These are all separate checkpoints. And then finally, I would also like you to tell me what are some other variables that the researchers should try to keep constant among their test subjects for the duration of the study, and give me at least 2. Now we're going to wind things down here for this lecture, but I think it's important that we step back and talk about what is the larger purpose of conducting all of this research. We've been down in the weeds talking about the details. Let's step back and ask, why do we do this? It may seem obvious, but the objective of science is to build reliable knowledge, because knowledge also lies on a spectrum. There's certain things that we are very uncertain about, and then there are other things that we are very certain about. Everything starts on the uncertain end of the spectrum, where it is considered a hypothesis. A hypothesis is a proposed explanation of a natural phenomenon that can be tested through experiments. On the other end of the spectrum is the most certain, most reliable knowledge, and these are called theories. A theory is an explanation of a natural phenomenon that has proven true across many tests and is accepted as fact. And I'll admit this can be confusing sometimes, because the way that we use the word theory in everyday language is different from what it means in science. In everyday language, we might use the word theory as if it was the same thing as a hunch or a guess. So you might say, I have a theory that my coworker Julie is the one who ate my lunch out of the fridge. That's not how the word theory is used in science. In science, it is the equivalent of a fact. Theories are the most certain knowledge that we can possibly achieve in science. And getting a piece of knowledge from hypothesis status on the uncertain end of the spectrum to theory status on the certain end of the spectrum takes decades, if not hundreds of years, and the efforts of many, many researchers, countless studies over and over again demonstrating that this knowledge is true. So here are some examples of important theories that have been developed in biology, as well as ones that you may have heard of in other scientific fields. So in biology, there is cell theory, which states that all living things are made of self-replicating cells. Gene theory, which is that organisms' traits are encoded in units of DNA called genes. Germ theory, which is the theory that certain diseases are caused by microscopic organisms that can invade and infect the body. And the theory of evolution, which is that over generations, species gradually experience genetic changes. In physics, there's the theory of gravity, Einstein's theory of relativity. In geology, there's the theory of plate tectonics. There's also the theory of heliocentricity. All of these pieces of knowledge are among the most reliable and most well-proven that have ever been developed by science. One final topic that we need to cover is how scientists go about communicating and publishing the work that they are doing. So the primary avenue for communication by scientists is scientific journals. which are collections of research papers within a particular subject area. They're like really boring magazines that contain articles written by scientists who conducted research. And in order to be published in a scientific journal, a research paper must have been peer-reviewed, which is a process in which at least 2 independent scientists look at the research and confirm that it is of a high enough quality to be put out there into the world as a publication. And the peer review process is typically extremely rigorous and has very high standards. Research papers that do not pass peer review are simply not published. And sometimes researchers will go through years of having their research rejected and then going back and trying to make the necessary adjustments to improve its quality, trying again to be published, and so on. So this is the quality control mechanism for science communication. A typical research paper, if you were to open up one of these journals and read one, includes an introduction section that explains the background to the research and why it's being done, then a method section that explains how it was done, and then lastly, a results and discussion section that explains what they found and why it is significant. And then finally, at the very end of the paper, researchers are obligated to disclose whether they have any conflicts of interest, which is that thing we talked about earlier, where the researcher might stand to benefit from the results coming out a certain way. So this is where you can learn that information about a study as well. And there are dozens of these scientific journals where researchers publish their work, but on this slide, you can see a few of the major well-respected ones. Nature is the name of one large journal. Cell is another. The one on the right is the Journal of the American Medical Association, which focuses specifically on medical research. So with that, we've come to the end of our detailed discussion of the second topic of the lecture, which is what is science. I hope you learned something new and you're ready to take this information and put it into action as we move through our biology learning this semester. Don't forget to turn in the answers to your checkpoints, and I will see you in the next lecture.